Bioinformatics for Infectious Diseases will consist of several different elements that work together to provide an experience, an immersive experience for anybody that would like to learn about the applications of bioinformatics for infectious diseases. So this will include interactive sessions like we're having today. It will point you to the right kind of self-guided study materials that you can use in between sessions to cover the topics that we don't have enough time to cover, as well as some practical assignments. So you'll actually be given curated datasets. You'll be able to perform analysis on our big data analysis platform that we'll introduce you to today, as well as some coding exercises so that you can explore, visualize, and analyze this data for biological interpretation. I will not be the only one speaking both in today's session, but also throughout the program. We actually have a number of experienced mentors that will be guiding and assisting you throughout this program. And that includes Dr. Harpreet Kaur, who is a OmicsLogic bioinformatics trainer and has a PhD in bioinformatics, particularly with a specialization in genomics and oncology. As well as Dr. Mohit Mazumdar, who's also here in the chat, and he is an expert in chemoinformatics, as well as the application or the relationship between sequence and structure. And I'm sure that he will mention a little bit about his background towards the end of today's session. And so my name is Ilya Brodsky. I'm one of the co-founders and the CEO of Pine Biotech, but I'm involved in every aspect of what our company does. And I'm particularly excited about this program because I think it's on everyone's mind today. Even people that used to be interested in oncology and neuroscience are now seeing the impact of infectious diseases on so many areas that we have been interested in before. And I think that it's impossible to kind of overlook the significance of what's going on. Not only are we living in the midst of a pandemic, we always hear in the news how there's new development. And so because of these developments, it's important to understand what we can do with the type of data that is available in such great quantities on the public domain, and how we can actually contribute by analyzing such data to extract meaningful insights from some of these data sets. So what can they tell us essentially? So this program called Bioinformatics for Infectious Diseases is going to focus on the relevant tools, methods, data repositories, and provide some examples from curated data sets where we can take sample data and analyze it together during these interactive sessions, point you to some project examples on the Learn OmicsLogic portal where you can register for free, and we'll be utilizing some of those examples and tutorials during the session so that you can master some key elements of analysis and visualization tools and hopefully apply some of these methods to a project of your own. So the objective of this program, and what we sincerely hope for everyone that joins this program, is that you will start and learn about something that is relevant to a research project that you would like to develop. Now, this program is going to last for a little bit longer than just the previous program that we completed not so long ago. That program was exclusively focused on the COVID-19 variants. There's some overlap between that program and this program, but we hope to widen the perspective so that we can look at sequence variation, evolutionary and phylogenetic analysis like we've done before, talk about functionality and protein structure using several different examples, and then also look at the transcriptomic data analysis so that we understand the host responses and potentially some of the immunology of these infectious diseases. So let me, for those of you that are joining with minimal background in virology or infectious diseases, kind of start from a little bit of a beginning. What are infectious diseases and how are they different from non-infectious diseases? So infectious diseases spread by pathogenic microorganisms, and those can include bacteria, viruses, parasites, and fungi. And you can see some of the examples here on the left side. So flu, HIV, malaria, Some of these are very different types of diseases. So they affect different tissues, they affect different types of cells, they have very different mechanisms of infection. But we call them infectious diseases because there are several characteristics about them that are unique. So for example, transmission is an important factor. The way that they replicate is going to be an important factor. And this is going to be different from non-infectious diseases that are not caused by a pathogen. So typically they are considered to be caused by environmental factors, nutrition deficiencies, lifestyle choices, and genetic inheritance. And when we speak about this approach of studying infectious diseases, some of the things that we will be focusing on are replication or colonization, cell death. So how does the pathogen induce the process of deterioration and death, as well as release of toxic, and as a result of that, how we can understand organ and tissue damage. So these are some of the important factors. And surprisingly, in maybe the past 10 or 20 years, the researchers have discovered that even though we might not consider some diseases to be caused by pathogens, some of the conventional diseases that we think of as non-infectious, like cancer and neurodegenerative diseases, might be associated with some of these factors. And one of the primary reasons for that is actually related to the way that our body responds to infectious diseases. And so to talk a little bit about the response, you can see here that, for example, bacteria, and you can see that it is being, the response is the ability of the body to kind of intercept, right? I don't know how to properly say that, but to intervene. And so when we speak about infectious diseases, we also have to consider the immune response to those infectious diseases, where this rapid ability of the pathogen to replicate and colonize different tissues is going to be important for, okay. Sorry, 1 second. It looks like somebody signed me out of this. Okay, so, okay, Mohit and everyone else, if you're trying to use my account, please stop that. I think I need it for my presentation.
Okay, Ilya. So actually the meeting with, there's another meeting with African team about machine learning. So
well, maybe tell them to use your account or something else, because we do need this for now.
Sure. Okay. This is my actually. So yeah.
Okay. Okay, so as a result, we need to consider the history of interventions to help address the spread of infectious diseases. And over the course of the years, there have been several important interventions at the public health level that have been very successful at reducing the load from infectious diseases and mortality. So you can see here the rate of infection and the years, and some of the important interventions that have been developed over the year, like the health departments and the public health component. Then you can see the first continuous municipal use of chlorine in water in the United States. After pandemics, especially, these novel interventions have been applied in a much greater way. And then you can see how it helped intervene with transmission of important infections, the use of penicillin and the development of antibiotics, as well as not so long ago, if you think about it, in the 1950s, the introduction of effective vaccination. And so the types of challenges that we are facing today, the reason why they are so unexpected is that we have a fairly successful history of applying our understanding of infectious disease pathogens to prevention and to treatment of infectious diseases. And so even though we had this high level of confidence that We understand the pathogens. We understand that the way they interact with the host can lead to disease, and we know how to address and prevent that problem. In the recent pandemic, of course, there has been this novel appreciation of a much more in-depth characterization of pathogens that cause these outbreaks, and the ability to translate our understanding about these pathogens to effective interventions. And so here you can see how the recent pandemic has really pushed the boundaries of what we know about intervention and treatment, essentially making COVID-19 one of the leading causes of mortality for pulmonary diseases that have been around since, for decades now. And so what that had led to is a greater appreciation of the role that bioinformatics can play in our monitoring, characterization of infectious diseases, the way that they spread, and how we can treat them effectively. And so here you can see a slide from the New England Journal of Medicine, where they describe some of the applications of genomic sequencing to monitor these outbreaks of infectious diseases. Now, one of the first infectious outbreaks that has kind of been a breakthrough moment for genomic sequencing was the outbreak of Ebola virus in West Africa in 2014-2016. During that time, for one of the first times in history, genomic sequencing was applied in a on a large scale to monitor and characterize the Ebola virus that was causing that outbreak. But you can see some of the other insights that could be gained from the analysis of that data, including insights into virulence, protein expression to understand potential proteins for drug design, proteins and immune response, diagnostics so that you can really understand the type of a strain or even a specific variant that is causing some particular aspect of the clinical manifestation of disease, insights into growth requirements so that you can understand drug susceptibility, specific sequences for strain detection and outbreak investigation. Potentially there could be zoonotic transmission or a rapid increase in transmission that could be associated with the genomic sequence. So really, genomics has become a central focus for some of the infectious disease monitoring and translational research. And that's why using a lot of these applications combined with the immunology of disease is really so centered around data management, data analysis, and interpretation. And so when we speak about bioinformatics for infectious diseases, some of the examples that we're going to be looking at is how can we compare different sequences from different similar viruses that are nevertheless divergent over time and causing different clinical manifestations? How can we characterize the molecular level of infectious disease so that we can compare the immune responses and the type of responses that the human body has to these pathogens? How can we identify some of the important regions on proteins that are so critical for the infection itself? And how can we incorporate our ability to manage and analyze data to integrate this information together and gain some reliable and interpretable biological insights into disease progression, transmission, and treatment of, specifically, we will probably focus on bacterial, viral, and parasite pathogens that cause some of the major diseases out there, like COVID-19, Ebola virus, malaria, tuberculosis, and others. So before we continue, I want to ask if there are any questions. So again, if you have any questions, please put them in the chat. I'm going to just give it one moment here for anyone to write it out. So the objective of this session today is to really make sure that the topics of the upcoming program are clear, that you understand how you might benefit from joining this program. And if you have any questions, either about the topics that I've covered so far or about any of these follow-up topics, please put those in the chat and I will address them either now, or if I do have something prepared already, I'll mention that as well. Okay, so if you have any questions, you can continue posting them in the chat. I will pause in a few more slides and address them as well. Okay, and so Arjun is asking, is there a certificate of completion? How valuable could a certificate of completion from this course be in application to a master's program? So this is a great question. I think that anyone that is trying to learn bioinformatics, one of the important part of mastering bioinformatics, I think, is to apply it to a project of your own. And likely you have a thesis or a project as a part of your master's. And that's one of the things that you can really focus on during this program. I don't think that people today, when they hire people from master degrees, are looking so much at the certificate that you will get. I think more so they will be looking for how knowledgeable you are to apply what you have learned to tackle a certain problem. And so we'll discuss several case studies in this program that will allow you to see what is the application of bioinformatics from beginning to the end of a research project. So how can you find a good data set? How can you plan your analysis? How can you complete that analysis? And what are some of the important critical questions, research questions that you can ask about the data set that you have found so that the analysis that you present addresses those questions in a comprehensive way. So you will get a certificate of completion, but again, I think the major outcome for anyone in a master's level will be to complete a project and demonstrate your understanding of that problem. So Arjun, let me know if I answered your question, and I'll be happy to expand on that later on. And then Lewis is asking, can I share the last two publications showed in the slides? Yes. So we will share all of the links for these publications after the session is over. If you could reach out to us by email so that we have your email address, and we will be happy to share that with you. Okay, and what does it take to complete a project? So we'll talk a little bit about the different stages of this program. As I mentioned, it's going to include training sessions and interactive sessions like we're having now. It will include some practical introduction to methods of analysis so that you can take curated data sets, process them, and make sure that you're getting the right results. But also at the end, we will leave some time for you to share with us a proposal on a project that you might find in the public domain. So data sets from NCBI virus, from GIS-Aid, from other types of public domain repositories like maybe Malaria Gen. So if you know of a data set, you will have plenty of time to find and kind of read the literature about that data set, propose a project that you describe just kind of based on your understanding of the logic. This is the data set, this is the question or a comparison that I will ask. Maybe you will perform some exploratory data analysis, and at the end, you'll have the opportunity to get some feedback on the project. And so you will be able to address any issues that come up. So you can maybe enhance your project, make it more comprehensive, make it more interesting, maybe make it more focused on the topics that are relevant in that process. Okay, so Abukari, let me know if I answer that question. Okay, great. So because we are living in the time of the pandemic, right? We probably all have heard about SARS-CoV-2, the spike protein, the genomic variation of that spike protein. And I think that, to get started with doing something meaningful in this particular pandemic, the first thing that we need to understand is how relevant the different data that is available to an impactful question, right? And so I think that I would divide this training into two parts. The first part, which you might as well do on your own, or you can do it together with our group here, is to learn about some of the basics related to where did this virus come from? How do we use bioinformatics to analyze data and test some of the hypotheses that were proposed? What are some of the basic outcomes of the analysis that we can use to answer some of the questions? And so I'm going to go through a few examples, and this is the first one, and I'm using it as the first one because this kind of an introductory project is something that you can try out after you have completed your registration on this portal. So again, I know that we already did this in the beginning of today's session, but if you have already logged in, let's go back to learn.omicslogic.com. And if you haven't, please register on this portal so that you can follow along and see some of the examples that we will be showing here in the next few slides. Okay, so this is the link. You will see that once you have logged in, you will have a profile and you will have some personal suggestions based on your progress. So as you start completing some of these tutorials and mastering some of these skills, it will start changing as you go through the coursework. And so right here, you see that we have the COVID-19 origin and pathogenesis of SARS-CoV-2. So I'm going to share that link with you as well. Again, if you haven't registered, registration here is free. So just please go ahead and get started. And one of the first things, and this I think will provide you with a good understanding of how this program will work, you will see setting the background, right? Setting the stage for what kind of questions should we be asking and how to translate those theoretical questions to practical data analysis process. So you'll see here some introduction about the coronaviruses and what do they do, what is the important part of it with transmission, and then you will also see what are some of the important factors that you can learn about here. And here you will see some of the objectives, some of the specific objectives that will be covered in this course, in this project. And so similar to this project, in the course of this whole program, we will actually go through quite a number of examples like this. So this one is on SARS-CoV-2. Another one that we will go through is malaria, where databases like MalariaGen and PlasmoDB offer us access to a comprehensive collection of data from different regions where malaria is endemic to identify some important factors on drug resistance, on transmission, as well as vaccine efficacy in application to different types of data about different proteins, different populations, different years. And so we'll be exploring that data together as well as a part of this program. As I mentioned, one of the first examples where pandemics or epidemics have been characterized at the genomic level almost in real time is the Ebola virus. And so one of the projects that we will be focused on is that tracking of Ebola virus evolution across that 2014-2016 epidemic in West Africa. And so as a result of these projects that we will dive into to understand their background, to understand the specific questions that could be asked about them, and understand some of the tools and methodologies, you will be able to appreciate the role of omics data in infectious disease research and the application of bioinformatics to be able to ask specific questions about these data sets. And so that will give you an insight into viral genomics and evolution, immune response biomarkers, and the relationship between microbiome and infection. Now, at the core of all of these types of omics data, we will primarily focus on next generation sequencing. And one of the aspects of next generation sequencing is that the technology itself has evolved rapidly over the past couple of decades, essentially leading us to where we are today, where there's just millions and millions of sequences and hundreds of thousands of data sets that are available for anyone to explore and access in the public domain. That means that as someone that comes without the lab capabilities of generating your own data, you can have access to data sets produced at top labs around the world in the best academic centers where people that have been trained for years are sharing data that they generate, and all of us can just take that data and analyze it. And what that means is that it eliminates the concerns about the quality of data. It eliminates the concerns about the quantity of data. So both quantity is there and quality is there. And so I think that makes this an exciting opportunity. And I think that infectious diseases is one of those areas where data is even more prevalent and available than some of the other conditions that you might be interested in. But to understand this data, we have to appreciate the heterogeneity of this data as well. So essentially, as I mentioned, the sequencing technologies have evolved over the years tremendously. And now we have short read sequences, long read sequences. Some of the technologies are much easier to generate quality data. Some of them have some of these other issues. And so we will review the different types of next generation sequencing data and data types before we jump into that. And so before I go into that process of next generation sequencing, again, I want to ask if you have any questions about what we have covered so far. So what we covered so far is the overview of the OmicsLogic Learn platform or portal, where you will have access to datasets and tutorials that you can learn from asynchronously. Number two, we started talking about infectious diseases and how research in infectious diseases can be conducted as a part of this program, where you will be able to apply the tools that you learn to data sets that you find on your own in the public domain. Number three, you will understand the different types of data that are available for the questions that you might be asking. So you really have the freedom to choose the type of a project or the type of an application or a question that you might have about these data sets. So again, if you have a question, please put that in the chat box, and I'll be, if I see the questions now, I'll be answering them now. If not, then we will wait until the next pause that we will have to answer any questions that you might have. Okay, so Dr. Amitabh is asking, is the best pathway to complete the introduction to Bioinfo course before enrolling for this one? Yes, if you are completely new to bioinformatics, what I would recommend, and let me actually show you a couple of things where you can get started, depending on kind of where you see yourself going with this process. There's a course on Introduction to Bioinformatics that explains the different applications of bioinformatics to biomedicine, to agriculture, etc. There's also a course called Bytes and Molecules. So Bytes and Molecules is a course for somebody who's just beginning to kind of learn about some of these topics. And you will see that here you will have an introduction to the Human Genome Project. You will see the application of citizen science. How do these two kind of paradigms converge together to create a need for bioinformatics or for training in data science? And then you will see some of the basics. What is the genome? What are nucleotides? What are amino acids? How do we go from sequence to function, right? So some of those basic things are going to be explained in this course as well. So either one, Introduction to Bioinformatics, is kind of probably the next step. And then Bytes and Molecules is the kind of first level where you would begin as a newbie to this topic. Does that answer your question, Dr. Amitabh? All right, excellent. So let's go on. So next generation sequencing. To appreciate the opportunities provided by next generation sequencing, we really need to understand how next generation sequencing works. And the way it works is that there's a machine that essentially takes the raw biological material, or RNA and DNA, and converts it into digital data. And this data consists of short sequences, or reads, as we call them, that now represent a digital snapshot of what happens at the subcellular level. Now, when we speak about infectious diseases, that means that we have millions and millions of reads about that pathogen, the virus, the bacteria, or the parasite, and potentially how does it actually look in a real biological sample. And it looks like this, right? So this is the result of mapping those short reads onto the reference genome. This is, by the way, one of the first examples of mapping the sequencing data from the Wuhan outbreak onto the reference genome of the previously known SARS-1 virus, where you can see how many, many short reads provide us with an understanding of what is the consensus sequence that we're ultimately going to be looking at. And so to be able to analyze this data, we can perform what's called sequence alignment. And the sequence alignment could be applied in two different ways. One of them is alignment to a reference when we have the raw reads and we want to understand the consensus or the variation from the reference. Or if we have a number of these consensus sequences, how do they relate to each other, or multiple sequence alignment, where we can now explore the relationship between them based on variation that we see in the sequences themselves. And so to see the application of how these methods could be used, let's go on to the server.TBioinfo, where we will see how we can analyze this data. And this is going to be for anyone, even if you don't have any background in bioinformatics at all. So I'm going to go to server.tbio.info. If you registered on the Learn OmicsLogic portal, you can use the same password that you used there. Maybe some of you already have a password, so just log in. And we'll take a look at one of the sections to kind of see what the process of analysis consists of. So once you're here, we will see how this portal has several different topics that you could apply analysis to. And that includes analysis of next generation sequencing data, mass spectroscopy, structural biology, as well as methods for data integration and modeling. And so what we want to do is see how analysis of these sequences can help reveal relationship between these sequences in application to a question that we might have about the SARS-CoV-2 virus. So I'm going to go under Phylogenetic Tree, and I'll find here several different demo examples. And one of those examples is going to be this one, SARS-CoV-2 origin, right? So it says I have to click on the dropdown, and then I have to click on demo SARS-CoV-2 origin. So as a demo, as an example, we don't have to upload the data. The data is already here. What we need to do is we need to click on several of these buttons to see how this data could be analyzed. So what is the process? What is the flow or the logic behind such analysis? So we have two options. As we mentioned, we can upload FASTQ or FASTA data. In this case, you can see the input files, and those are bat coronavirus, bat SARS-CoV-2, human SARS-CoV-2, as well as some pangolin datasets on SARS-CoV-2, right? So the question here, and this is the question asked in the tutorial that I shared earlier, is what is the relationship between different sequences? How could they be organized into what's called the phylogenetic tree so that we can analyze their evolutionary closeness, how closely related they are, and when could they have diverged into these separate directions of adaptation? And that could help answer the question, what is this virus in humans, and how could it be related to some of these proposed intermediate hosts like bats or pangolins? So I'll click on start. Then I'm going to use multiple sequence alignment to compare these. I will convert them to amino acid sequences so I can understand the impact of those mutations on the protein sequences. And then I will use all phylogenetic methods that are listed right here to perform this phylogenetic analysis. Now, once I have many different phylogenetic trees based on different models, mathematical models that kind of represent the molecular clock or the molecular evolution that I observe at the sequence level, I can also analyze the phylogenetic tree structures to identify the best one that is kind of convergent between these different trees that I've gotten as a result of many different methods of analysis. So after I'm done with this pipeline, I will have here several different outputs. And I'm going to take one of these to see whether I can understand something about the relationship of these viral genomes. So when I open this plot, I'm going to see here different names, and these names represent different FASTA files that I have uploaded. And so to appreciate the results, I have to annotate them. So essentially, I have to look at how are these represented as a phylogenetic tree, right? So each branch here represents a split from the origin, and the nodes represent a common potential ancestor. And so we can use this analysis to understand how evolutionary time or adaptation, the process of evolution that we observe at the genomic level, has gotten us the present day species that we have collected, the samples that we have collected. So if we apply this technique to Ebola virus, we can ask the question about how do different regions like Gabon, Liberia, and Great Britain relate to each other based on genomic sequences. If we look at the SARS-CoV-2, we can also identify, as I mentioned, the relationship between the human SARS-CoV-2, the bat coronavirus, RAG13, or the bat SARS-like coronavirus, as well as the pangolin and other types of SARS human viruses. So we can see the relationship overall. We can also find specific amino acid mutations in critical regions. For example, this spike glycoprotein mutation, D to G, that was prevalent and became one of the dominant mutations, potentially linked to increased transmission rate in the SARS coronavirus. So that is the second part of what we will do in this program, and we'll talk about how do we find what are called mutations or SNPs. And so these SNPs or mutations or variants are important to be able to analyze, categorize, and characterize in terms of their impact on the phenotype. And so once we perform alignment, and we can perform alignment on nucleotide and amino acid level, what we're interested in are different types of mutations: missense mutations, nonsense mutations, and frameshift mutations. And we want to find particularly those positions that could indicate a change in the protein structure. So it could be done using multiple sequence alignment, or it could be used by looking at all of the reads that we have that represent potentially many different viruses present in a sample, and identifying variants of importance. And that is called variant calling. And so by variant calling, we can identify those SNPs that have a potential impact, and then we can use different methods of characterization of protein function to understand how relevant they might be for a specific question of the impact of that mutation on a structural component that we have identified as important to what we're studying. And so here you can see several different ways that we can look at that, and several different ways that we can interpret this kind of information. So what we'll do in the program itself is we'll actually take these models of proteins that are on the host side. Like, for example, here you can see the host side ACE2 protein, which is in blue. But in reality, this protein is actually part of the complex, and this complex sits within the BO81 sodium-dependent neutral amino acid transporter. And then we'll also find the spike protein, which also what we have from crystallography studies is just a component of that protein, but we'll look at how they relate to each other by using UCSF chimera and kind of mapping those 3D structures onto each other, helping us understand how this virus actually binds to the host cell. You can see the membrane right here, and then try to make the interpretation of a mutation, of a change in the structure based on the impact that that would have on protein-protein interaction, just like we're seeing here. So again, this will give us some insight into how spike protein of SARS-CoV-2, which is a critical protein for both the transmission, but also the vaccine prevention of that transmission and prevention of disease, play a critical role. The mutations, right, those variants play a critical role in all of those aspects as we anticipate this pandemic to continue and spread. And so in this program, the Bioinformatics for Infectious Diseases, we will cover sequence variation, evolutionary relationships and how those are measured, functional inference for sequence to structure and function, as well as host response. How can we analyze the response of the host, transcriptomic data, RNA-seq data, to find patterns that can explain some of the reactions that we see between mild maybe severe disease, maybe symptomatic disease, right? Maybe we can look into the immunology of different immune cells, etc.